From structure to state, why biology may need a soft matter framework. Modern medicine has achieved something astonishing. It has learned to look inside the body with extraordinary precision and to translate that vision into treatment. It can identify cell types, map signaling pathways, measure biochemical markers, track hormones, visualize organs, detect molecular change, and intervene with a sophistication that would once have seemed impossible. That success is real. It has given us life-saving surgery, transplantation, emergency care, imaging, pharmacology, and a level of diagnostic reach unmatched in human history.
Yet every successful way of seeing also becomes a filter. The more powerfully it explains one class of phenomena, the more easily it can become blind to another. A framework that works brilliantly in one domain can begin to feel universal. Over time, its assumptions become so familiar that they are no longer experienced as assumptions at all. They harden into common sense. At that point, the question is no longer whether the framework is useful, but whether it has become too narrow for the territory it is trying to govern.
That is where the argument for a soft matter framework begins. The issue is not that modern biology is wrong, plainly it is not. The issue is whether the dominant medical model is complete enough to deal with all the behaviours that living materials can display. Modern medicine has largely been built around stable structures, discrete parts, identifiable mechanisms, and measurable endpoints. It asks what a thing is made of, what pathway it belongs to, what receptor it activates, what marker it produces, what lesion it forms, and what category it fits into. Those questions are often exactly the right ones. But they are not the only questions available. There are situations in which the most important feature of a system is not simply what it is made of, but how it is organised, how it responds to local conditions, what state it is in, and how that state changes over time.
That distinction becomes especially important when one is dealing with complex fluids and responsive biological media. Blood is the most obvious example. In ordinary medical language, blood is a transport fluid. It carries oxygen, nutrients, hormones, waste products, immune cells, platelets, proteins, and a host of dissolved molecules. This description is accurate, but it is not complete. Under certain conditions, blood behaves in ways that are not well captured by the language of transport alone. It can separate into domains. Boundaries can emerge. Gel-like regions may appear. Fibers may develop. Particulate and vesicular patterns may arise. Different regions of the same sample may follow different trajectories. Structures may appear, dissolve, reorganize, or persist while neighboring material continues to change. At that point, one is no longer looking only at a collection of components. One is looking at a dynamic medium.
This is where soft matter becomes useful. Soft matter is the domain of materials that are neither rigid solids nor simple liquids. It includes gels, colloids, emulsions, vesicles, polymers, thin films, and liquid crystalline phases. These materials are not defined only by composition. They are defined by organisation. They respond to local conditions. They form interfaces. They separate into phases. They occupy transient but structured states. They transform under shifts in temperature, hydration, confinement, flow, concentration, or field conditions. In such systems, a boundary is not just an outline, it is an active interface. A gel is not merely an object, it is a state. A particulate field is not necessarily residue. It may be part of a trajectory. Structure and process begin to overlap.
This matters because biology has tended to inherit a strongly object-based view of the body. Cells are treated as discrete units. Molecules are treated as the primary drivers of function. Pathology is often framed as deviation from a stable norm. Diagnosis frequently rests on identifying structural abnormalities or chemical imbalances at a particular moment in time. That approach is immensely productive when the phenomena of interest are stable, repeatable, and reducible to components and pathways. It is less strong where organization emerges dynamically, where boundaries shift, and where the behavior of the system depends as much on state as on substance.
A soft matter framework does not tell us that chemistry stops mattering. It tells us that chemistry may not be enough. The same components can behave differently depending on how they are arranged. Boundaries, interfaces, local geometries and phase relationships can shape behaviour profoundly. A system may contain the same molecules yet organise itself in different ways under different conditions. In that sense, organisation is not secondary to composition. It is one of the ways composition becomes active.
That distinction may sound abstract, but it becomes very practical as soon as one moves from fixed specimens to time-based observation. One of the deepest habits of modern medicine is the snapshot. A blood test captures a moment, a pathology slide captures a moment, an imaging scan captures a moment, a stained specimen captures a moment. These tools are indispensable, but they privilege endpoints over trajectories. They tell us what is visible now. They often tell us much less about how it formed, how stable it is, what conditions support it, and what it may become if those conditions shift.
This is especially important in microscopy. A still image can be compelling, but it can also be misleading. A structure seen at one moment may be transitional rather than final. It may be part of a pathway rather than an endpoint. It may be a transient state, a metastable state, or a regional expression of a broader field dynamic. A static image may therefore encourage premature labelling. Something is called artifact, fibrin contamination, a clot, a crystal, a residue, pathology, or nothing of interest. Once that label is applied, interpretation begins to harden around it. Perception narrows. Downstream inference becomes governed by the first convenient name.
A soft matter framework offers a more disciplined alternative. It allows one to stay with behavior before forcing identity. Instead of asking only what this structure is, one can ask what it is doing. Under what conditions did it appear? Did it persist? Did it reorganize? Did it remain separate from neighboring material? Did it dissolve? Did it recur in comparable preparations? Did it arise within a boundary-defined domain? Did it emerge from a gel-like phase? Did it remain coherent while surrounding material continued to transform? Those questions do not avoid rigor. They demand it.
The concept of metastability becomes crucial here. In ordinary thought, we divide things into stable and unstable, present and absent, normal and abnormal. Soft matter complicates this. A system may occupy a state that is stable for a time but still poised for rapid change. Something may persist under one set of conditions and reorganize under another. A form may be neither permanent nor random. It may be conditionally stable. That matters enormously in biology because it opens a different way of thinking about observed forms. Some structures may not be fixed abnormalities or isolated curiosities, but expressions of a system in transition.
This does not mean that diagnosis becomes impossible or that pathology categories stop mattering. It means that a second layer of description becomes necessary. A blood film or pathology report may remain valid, but it may not exhaust the behavioral possibilities of the material system being examined. Composition and structure are one layer. Organization, state, and transition are another. Modern medicine is very strong in the first. It is less developed in the second.
The argument becomes sharper when one considers interventions. Pharmaceutical and injectable materials are usually described in terms of composition, dose, delivery, and intended biochemical effect. That is exactly as it should be, but it may not be the whole story. If the receiving medium is itself dynamic and multi-phase, then additional questions become reasonable. How does introduced material distribute within that medium? Does it remain dispersed or aggregate? Does it interact with boundaries, interfaces, or particulate fields? Does it occupy metastable states? Does it alter local organization? Does the state of the receiving medium influence its behavior in ways not captured by composition alone?
These are not hostile questions. They do not deny immunology, pharmacology, or molecular biology. They widen the investigative field. They remind us that a biological intervention enters not a passive container, but a dynamic environment. The body is not just a bag of targets. It is a material system with organization, responsiveness, and trajectory. The behavior of what is introduced may therefore depend not only on what it is, but on the state of the system it enters.
This is one reason why soft matter may offer something more robust than many of the broader umbrella phrases that already circulate in medicine. Terms such as integrative medicine, systems biology, systemic thinking, and complexity theory all point in the direction of wholeness and interconnection. They arise from a legitimate dissatisfaction with overly reductionist thinking. They recognize that the body is not merely a collection of isolated parts, but they often remain too broad or too abstract to do the descriptive work required when one is dealing with actual material behavior.
Integrative medicine can easily become a philosophical posture rather than a precise framework. It often gestures toward the whole person, the interdependence of systems, and the limits of symptom-based treatment. All of this may be true and valuable. But when one is looking at a changing blood field, a gel-like domain, a boundary-defined interface, or a time-dependent structural transformation, integration does not tell us very much. It does not specify phase, interface, metastability, confinement, reorganization, or the conditions under which one state becomes another. It is a broad orientation, not a detailed descriptive language.
Systems biology is in some ways more rigorous, but it too has limitations here. It tends to focus on networks, signaling, interactions, and large-scale computational modeling. It is strong when the relevant complexity can be formalized in terms of genes, proteins, pathways, and regulatory loops, but it often remains composition-heavy and mechanism-centered. It is less naturally equipped to describe emergent material states in terms of gels, boundaries, colloidal behaviour, phase separation, and medium-level organisation, unless those things are already translated into variables it knows how to handle. In practice, it can remain highly sophisticated while still missing the direct physical behaviour of the material field. Complexity theory faces a similar problem. It is useful in naming nonlinearity, emergence, self-organization, and sensitivity to initial conditions, but it often stays at a high level of abstraction. It can tell us that systems are complex without telling us what the material transitions actually are. It can describe pattern in general while offering relatively little concrete language for what is happening in a specific fluid medium under specific conditions. It risks becoming a way of sounding profound while remaining descriptively thin.
Soft matter, by contrast, is robust precisely because it is neither vague nor purely metaphorical. It gives operational terms: phase separation, interface, boundary, gel, vesicle, colloid, metastability, confinement, reorganization, thin film, coupling, transition. These are not merely holistic sentiments, they are material descriptors. They can be observed, compared, challenged, sequenced, and tested. They allow one to move beyond the bland claim that everything is connected and ask instead how connection is physically organized in a given medium.
That is why soft matter may be more useful than the softer rhetoric of integrative thinking and more grounded than the grand abstractions of complexity language. It sits closer to the behaviour of matter itself. It is not merely a philosophy of interdependence. It is a disciplined account of how organised materials behave when they are not rigid, not simple, and not static.
This also helps explain why certain observations have historically struggled for recognition. There have long been observers, often at the margins of orthodox medicine and biology, who describe dynamic structural behavior in blood and related fluids. What they often lacked was not necessarily observational seriousness, but a framework that mainstream science could accept. Their descriptions of emergence, reorganization, and disappearance became entangled with explanatory systems that directly conflicted with dominant biology. Once that happened, observation and interpretation were judged together. If the explanation seemed untenable, the entire body of work was easily dismissed.
A modern soft matter framework allows a more precise distinction. It makes it possible to say that dynamic structural behaviour may indeed have been observed without endorsing every theory built around it. Some historical claims may remain unsupported or implausible, but that does not mean the observed transformations were imaginary. It may mean that they were interpreted through the wrong conceptual lens. A behaviour that was once described as transformation between discrete living forms may look very different if redescribed in terms of phase behaviour, metastability, boundary-dependent reorganisation, or structural change within a continuous medium.
This distinction matters in the present as well. Scientific communities rely on shared frameworks. They evaluate new findings not only by evidence, but by how that evidence is framed. If a phenomenon arrives in language that conflicts sharply with prevailing models, it is more likely to be dismissed, even when there may be something real in the observation. Conversely, if the same phenomenon can be described in terms compatible with accepted forms of evaluation, it has a greater chance of being examined seriously. This is not proof of censorship or bad faith. It is a structural feature of how knowledge stabilizes itself.
That is why clear language matters so much. Without it, one gets two equally unhelpful outcomes. Either an observer leaps into overstatement and buries the signal in extravagant claims, or the observer remains too vague and cannot preserve the signal at all. A structured framework such as soft matter helps by separating observation from interpretation. It allows one to state carefully what is visible, what changes, what persists, and what remains unresolved. In that sense, its value lies not only in what it explains, but in what it allows to be clearly said.
There is also a broader lesson here about institutions. Scientific and medical frameworks are embedded in training, funding, publication, regulation, and professional culture. They are not abstract systems floating free of history. They are sustained by infrastructures of legitimacy. When a framework has delivered practical success, those infrastructures naturally favor continuity. That is usually sensible. But it also means that unfamiliar observations may struggle to gain traction if they do not fit established categories. A field can become so good at seeing one class of phenomena that it becomes less responsive to another, not because the latter are unreal, but because there is no stable way yet to integrate them.
Soft matter may therefore matter as much for epistemology as for biology. It raises the question of what counts as a legitimate object of knowledge. A structure-based framework seeks stable entities and repeatable classifications. A state-based framework is more comfortable with emergence, transition, partial coherence, and dynamic organization. Both approaches have value. The challenge is to let them coexist without one automatically cancelling the other.
A richer biology would not abandon cells, molecules, pathways, receptors, and diagnostics. It would simply become more sensitive to media, interfaces, local conditions, temporal behavior, and the possibility that some biological phenomena are better understood as evolving states rather than fixed objects. Such an expansion would not weaken medicine. It would enlarge it. It would allow medicine to ask not only what a material is made of, but what it is doing.
This would also restore a particular kind of humility, not the humility that says nothing can be known, not the humility that retreats from difficult observations because they are controversial, but the humility that recognizes that successful models may still be incomplete and that seeing more may require describing differently. That is a far more demanding humility than fashionable uncertainty. It does not lower standards, it raises them. This is especially important in an era of increasingly complex biomedical interventions. The more medicine works with structured preparations, delivery systems, engineered particles, and responsive tissue environments, the less adequate a purely static ontology may become. If the medium matters, then state matters. If organisation influences outcome, then organisation must become part of the description. If transition governs behaviour, then time-based observation becomes indispensable.
The broader significance of this shift goes well beyond microscopy. It affects how we think about intervention, response, evidence, and regulation. It changes the kinds of questions that count as serious. It suggests that certain forms of dismissal may reflect not the weakness of the phenomenon, but the weakness of the vocabulary available to describe it. And it offers a way to help observers articulate what they are seeing without forcing them into either overclaim or silence. The central proposal, then, is both modest and far-reaching. Biology may need to think in terms of both structure and state, both composition and organization, both snapshot and trajectory, both local form and field behavior. It may need to become more careful about the difference between what is directly observed and what is inferred. It may need to revisit some historically marginal observations without inheriting all the theories once attached to them. And it may need to accept that some systems are not best described as simple collections of parts, but as responsive media whose organization is itself biologically consequential.
None of this requires discarding the achievements of modern medicine. It requires expansion, not replacement. The body is still made of structures, but it may also be a world of states. Blood is still composed of cells and plasma, but it may also be a dynamic soft matter system. Pharmaceutical materials are still defined by composition, but their organisation and behaviour may matter more than standard training has prepared us to see.
That is why the move from structure to state matters. It does not ask biology to become less rigorous. It asks it to become more observant. It does not ask medicine to abandon what it already knows. It asks whether, at the edge of its current success, it may be ready to notice a wider material reality than the one its dominant categories were designed to hold.
The immediate task is not to force certainty where it does not belong. It is simpler and harder than that. Observe carefully, describe precisely, follow change over time, resist premature labelling, separate evidence from overreach. Let complex living materials show their behaviour before interpretation closes too quickly around them. That is not a rejection of science, it is a demand for better science.